This is the solutions video for problem set number 11 for Kevin Ahern's BB450 class at Oregon State University. In this problem set, I'll be working problems relating to the phenomenon of signaling as has been discussed in class. The first question asks, chemical derivatives of GTP are available that contain a sulfur in place of one of the phosphates of GTP. The resulting molecule acts like GTP, but cannot be hydrolyzed. Predict the effect of these molecules in a cell where the beta-adrenergic receptor is activated. Well, we remember, of course, that the beta-adrenergic receptor's activation results in the conversion of the alpha subunit from containing GDP to containing GTP. Now, I'm going to label the modified form of the GTP as GTSP. And that GTSP will bind to the alpha subunit just as the GTP will, but because it cannot be hydrolyzed, the alpha subunit will remain in the active state because you remember that this is the active form of the alpha subunit. Well, that active form of the alpha subunit means that this is the active form as well, but the big difference between the two is that this can be inactivated by hydrolysis. This can't be inactivated. Well, since the alpha subunit plays a role in the kinase cascade, and the end product of the kinase cascade is to activate the breakdown of glycogen, cells that were treated with this compound would therefore break down all of their glycogen and would have no stores left available to them. Question number 2 says, a mutant liver cell is found that has an excessive amount of phosphodiesterase. Predict the way in which this cell would differ from normal liver cells. Well, we remember from class that phosphodiesterase is important for converting cyclic AMP to AMP. Phosphodiesterase. Okay, so why is that important? Well, we remember that protein kinase A, which I'll call pKa, is activated by the presence of cyclic AMP. And cyclic AMP is produced in response to the binding by the beta-adrenergic receptor of its appropriate hormone, either epinephrine or glucagon. If the cell had an excess amount of phosphodiesterase, then the cell would have very little cyclic AMP. So in the absence of cyclic AMP, protein kinase A is going to be inactive more often than not, meaning that the kinase cascade will be inactive, and consequently, glycogen breakdown, it's a C, will also be inactive. So the consequences of an overactive phosphodiesterase would be just that. The last part of the question says, predict the way in which this cell, I guess I've done that. The question number 3 says, how might the mutant cell in the previous question be treated to make it act more like a normal liver cell? Well, one way to do that would be to treat the liver cell with an inhibitor of it. And one of the inhibitors of phosphodiesterase that I mentioned in class was caffeine. Caffeine inhibits phosphodiesterase. And the appropriate amount of caffeine that was used to treat the cell might be able to reduce the level of phosphodiesterase activity back down to a normal level such that the cell could respond appropriately to hormones as necessary. Question number 4 says, another mutant liver cell is found that has a mutant alpha subunit of a G protein that is not activated by binding of GTP. Predict the way in which this cell would differ from normal liver cells. So if we had a liver cell that had an alpha subunit that bound to GTP but did not become activated by it, what would happen? Well, if this did not become activated, it would not go and interact with adenylate cyclase. So this interaction would not occur. And adenylate cyclase would not make cyclic AMP. Cyclic AMP would not activate protein kinase A. Protein kinase A would not activate the kinase cascade. And the kinase cascade would not activate glycogen breakdown. So this cell would not be able to go through all these processes and would not be able to provide glucose to the body as needed. The last question says, a mutant cell is found which contains an insulin receptor that cannot autophosphorylate. Predict the behavior of this cell compared to an unmutated cell. Well, we remember that the autophosphorylation of the insulin receptor, the insulin receptor is a receptor tyrosine kinase, and that receptor tyrosine kinase is activated by its autophosphorylation of its individual tails. Without the autophosphorylation, there will not be binding of the signaling complex to the insulin receptor, and in the absence of binding to so if we have no signaling complex, so this would not occur. The signaling complex, in turn, would not go through the series of steps necessary to activate the movement of GLUT4 to the cell surface. So that would not occur. Which in turn would mean that the cell would be unable to move glucose into it. This would, of course, be very drastic for the cell. This would not occur. That is, it's not able to move glucose into the cell. Therefore, what we would see is that blood glucose levels in an individual with this problem would likely be very high, and the cells themselves would not be very healthy because they would not be able to obtain the glucose that they needed, at least not through the GLUT4 receptor.